For several decades, the International Council for Harmonization, or ICH, has had a set of guidelines designed for development, maintenance, and validation of analytical procedures for assessment of the quality of drug substances and drug products. Recently, our revision has incorporated principles of analytical quality by design into their guidelines. Analytical quality by design, or AQbD, uses product and process understanding to develop analytical procedures that deliver results that meet predefined objectives. By integrating AQbD into the ICH guidelines for analytical method development, the analyst must arrive at a statistically relevant solution in developing their method. When it comes to arriving at the proper solution to method development, it requires a solid knowledge of the chemistry of the molecule, as well as an array of columns and mobile phases. The importance of method development in chromatography is best distilled in ICH Q14, which states, "The goal of development is to obtain an analytical procedure fit for its intended purpose, to measure an attribute of the material with the needed specificity, selectivity, accuracy, and/or precision over a reportable range." Method development is a cornerstone to drug development, as analysis methods via chromatography can provide a wide variety of information that assess safety and efficacy of a drug candidate, purity, size, and post-translational modifications. Therefore, an analysis method must have specificity, accuracy, and precision in measuring the molecule. Method development does pose its challenges, and broadly, there are two types of challenges that an analyst will face. The first one is the setup of method files, and that includes hardware and solvents that are used to acquire data. There can be plenty of combinations of columns and mobile phases that you'll use in setup of method files, taking you into the even hundreds for a small study. The second challenge is analyzing the huge amount of acquired data. It is tedious, prone to mistakes, and different people will do it differently. So one of the first challenges in method development is construction of method files. As I was talking about earlier, say you're developing an analysis method where you're assessing 6 different columns using 6 different mobile phases across 4 different gradients. That's already 144 method files, which will take hours to make. Automating this aspect of method development would be ideal by selecting a full factorial design of experiments. The goal of design of experiments, or DOE, is to shorten the time it takes to reach a solution, and that's partly achieved by reducing the number of runs. For example, if we want to determine the best combination of mobile phase concentration, flow rate, oven temperature, and gradient time, we need to set 3 levels for each parameter. And if we conduct the analysis using a full factorial design, which is a design for a total experiment, 81 analyses would need to be required. And if we assume 10 minutes per analysis, that analysis would, assuming each one takes 10 minutes, it would take 14 hours to complete the entire analysis. However, if you approach it using a box Bankin design or a central composite design, you can reduce the time of data acquisition to 1/3 of how long it would take using a full factorial. An efficient way to spend your time on method development should have an increase in automation, efficiency, and output. Approaching method development with AQbD principles requires the analyst to follow set guidelines to arrive at a statistically relevant solution. An analysis software for method development should equip you with the tools to analyze your data in complex ways that help you arrive at a statistically relevant solution, like evaluating each chromatogram based on the number of peaks and their resolution values, and ranking each method accordingly. It's also important for your analysis software to be capable of creating a design space and searching for the most optimum analytical conditions for your method. Even if this condition is not one of the analyzed conditions, the software should be able to determine that using the design space. Here, I've shown a design space for the ratio of acetonitrile in mobile phase and oven temp, but it can be created using any 2 parameters. On the right side, I've shown how other parameters can be used to identify areas within the design space where certain conditions are met. They can be symmetry factor for the peaks, last peak retention times, minimum resolutions, the list goes on and on. The software should be able to shade out the areas of the acceptable limits for these parameters, and the hatched area provides the space where these parameters are met. Your software should also be able to compare chromatograms of each compound on a chromatogram across several methods being ran. It's often difficult to keep track of peaks as the shape and retention time can change between methods. Therefore, it would be ideal if your software could automatically track those peaks using either their UV spectrum of the compound or by the mass to charge ratio if you're using a single quad mass spec, leading to even more specificity than UV profiles. Now, I've been posing a lot of ideas about what method development analysis software can do, but I'm going to tie it back to regulation. In ICH-Q2 R2, a guideline for analytical procedure development, they acknowledge that there are differences that often exist between various compendia and documents of the ICH member regulatory authorities, and they're meant to be bridged. When doing method development through an AQbD approach, an analyst's experience will often dictate how they interpret the regulatory guidelines when developing the method, and they'll approach the data when landing on an analysis method, building those aforementioned bridges on their own accord. Now, what if I told you that Shimadzu has its own offering of a software that builds these bridges for you? LabSolutions MD is meant to be your bridge builder by automating creation of method files, tracking peaks of importance across method candidates, and optimizing analysis conditions, regardless of an analyst's experience with method development. To further expand upon these capabilities, I'm going to hand it over to Brian Demansky, Strategic Collaboration Scientist at Shimadzu Scientific Instruments. Take it away, Brian.
So let me start by saying thank you to Kevin for his wonderful explanation of why we would use LabSolutions MD. My name again is Brian Demansky. I'm the Strategic Collaboration Scientist for Pharma and Biopharma at Shimadzu Scientific Instruments, and I'll be looking here at the applications and the data that's acquired and how we can analyze it using the LabSolutions MD to improve our method development process. And we'll look at a number of different use cases for the software and see how it's implemented as well from a hardware perspective. The process of creating an LC method can be broken down into three essential steps that are common in the method development arc. The first being screening, the second optimization, and following an optimized method being determined, evaluating the robustness of that determined method. In the screening process, we're often looking at multiple mobile phases and columns and trying to find a proximate answer. Once we have an acceptable separation, that separation then is improved and optimized in order to make it robust enough to survive the next stage of analysis. Once we've established a method that will be used for routine assays, we then need to see what will happen if we make small deviations to the assigned parameters. If mobile phases are not made quite correctly, if temperatures deviate from their original intended values, or if pumps or other portions of the system do not perform as intended. We want to know how far can we stray from those ideal conditions before we are out of specification. When using LabSolutions MD to engage in method development, the first portion of the software that the user will engage is the creation of the analysis schedule. Within this user interface, the user needs to select by checking each of the predefined mobile phases from the mobile phase database that they wish to use. They can then also check the box for each of the columns that they wish to screen the mobile phase conditions against. The column database allows for parameters such as pressure limitations and temperature requirements for columns to be considered, as well as solvent compatibility. The user then enters the sample information. Single or multiple samples can be accommodated. Single samples can be presented in one container or in multiple containers. And then the analytical conditions are specified. Detector parameters come from a base method file, which is then shared for all of the analysis, and gradient conditions are specified on screen or can be sourced from the method file. Gradient conditions can be set as isocratic or linear gradients, or can also be sophisticated stepwise gradients. Once that analytical information is entered, then the user selects the design of experiment methodology. Once that has been locked in, All of the batches are automatically created, including all required system and column equilibration steps, so that those things are not needed to be considered by the user. All of the equilibration steps will appear as lines in the batch. However, the data files that correspond to the equilibration data are automatically stored in a separate folder from the project data. When using LabSolutions MD, that ease of setting up your screening comes whether you're using LC or SFC. There's the flexibility of the software to accommodate a variety of different system configurations, as well as a number of different combinations of solvent choice. So when using LC, the Nexera series method scouting system enables the handling of up to 8 different mobile phases and up to 12 columns at up to 130 megapascal, or about 19,000 psi. What this does is eliminates the user's need to ever grab for a wrench or to take any steps that remove the automated nature of the system in terms of selecting solvents and selecting columns. When we up the ante and look at the SFC implementation within LabSolutions MD, we have the capability not only to do that same column and modifier selection in the SFC realm, but we have the ability to control the UHPLC SFC switching system. And we can, in an automated sense, accomplish both SFC and LC separations within a single or multiple enqueued batches, including changeover of conditions within the system. And while solvent switching valves as part of a method development system are not necessarily revolutionary, one thing that Shimadzu's Nexera series provides is the ability to do what is called solvent blending. We then use a low-pressure gradient valve in each of our installed pumps. If we're working with an LC or UHPLC system, then in both the aqueous and organic pump, we can have automatic proportioning valves that will allow us to create blends at a percentage level automatically. This can allow us to do things like prepare different pH levels or test different concentrations of small amount additives. Rather than having to make each individual level of buffer that we might like to test, we can make a strong solution and have a weak solution on the same valve and create proportions in any level we desire. This can then be automated in the software, and each of the construed solvent blending combinations is stored as an individual solvent in the solvent database and can be recalled at any later time. So let's look at a use case of how we might implement that solvent blending feature. In this case, we're using solvent blending to create a sodium phosphate buffer in a pH range from 2.5 to 8.5. In this case, we can see then the resulting chromatograms at each pH level. And when we apply our criteria for our ranking of the chromatograms and determine the evaluation value, we see that the optimum is at 7.5 pH. As you can see, by enabling the features of peak tracking and looking at the data from multiple points, we can follow where each of these species makes a transition in its ionization state due to the pH of the mobile phase. In addition to things like the evaluation value metrics, the ability to visualize the results that are obtained from the analysis schedule are key to the functionality of LabSolutions MD. If we're comparing, say, the results between 6 different columns that we've screened, we can, at a glance, see that column A in this instance produces the largest area of red within the investigated conditions and the predicted conditions. Red indicates an area of either optimal evaluation value, resolution, or peak count number. Those are the typical visualizations that are available at a click. The more blue we see, the less optimal that space is. In fact, we may get no separation in those spaces. So in this example, column D would not be one that would be a likely choice. Columns E, F, B, and C do have some regions where we see that red color indicating good separation, but we always want to have as large of a space covered by good separation as possible. That comes to robustness, as we'll discuss soon. Let's look at another use case where solvent blending becomes an important feature. When we think about the development of methods for oligonucleotide separations, oftentimes there are additives used, either ion pairing agents or things like hexafluoroisopropanol, that are not the easiest solvents to work with. They oftentimes leave behind residues on systems or have challenges with compatibility with flow path parts and materials. So our ability to minimize our separation time while achieving the desired level of separation and minimizing these components is key. And the optimal value for those small amount additives may be somewhere in between the classic 5, 10, or 20 millimolar preparations that you might do if you had to prepare them by hand. By allowing solvent blending to automatically prepare those for you, you can find much more accurately the minimum amount of those additives that's required to achieve an acceptable separation. And again, we can use the evaluation tools built into LabSolutions MD to not only obtain analysis, but to evaluate results and to see which of the chromatograms produces the best result. Looking further into the subject of oligonucleotides, we see that LabSolutions MD and its incorporation of peak tracking along with single quad mass spec can be particularly useful when trying to do impurity analysis or preparing analytical methods that will detect small level impurities in specialized synthetic oligonucleotides. Given the close relationship to the target compound, being small variations or substitutions, it's oftentimes a very challenging separation to get apart some of those minor component impurities. When we allow for the use of LabSolutions MD, we don't have to run every possible combination of conditions to try and target the ideal condition. We can use the concepts introduced by core composite design to choose points and interpolate between. And when we do that, we can then visualize our design space resolution and choose an optimized chromatogram that will be in the center of one of the high separation areas, so that if we do have subsequent changes to environmental conditions or instrument integrity, we still can achieve the separation needed when those changes are small. In an effort to constantly improve the LabSolutions MD product, one of the recently added features is the ability to visualize the peak to valley ratio. When we consider that in the use case for monoclonal antibody separations, we can see in this example that we have aggregates, the primary monomer, as well as fragments that are not well separated from the intended monoclonal antibody. As we look at the ratio of the valley and in between peaks B and C, we wish for that to be maximized to allow for the fragment to be separated from the pure monomer in order to achieve a purification. Seeing the space in which that accomplishes the greatest amount of separation is quite useful and can help to predict additional conditions that may be more favorable even still to encourage that separation. In the example workflows we've looked at up to now, the design spaces and the visualizations have been constructed around a screening gradient. Oftentimes, a simple screening gradient will accomplish the desired separation. But what if we have multiple components in a separation and we have very specific needs to resolve a certain critical pair or to get a specific pattern to the separation in order to accomplish purification. These are then challenges that we can subject to another portion of LabSolutions MD, namely the automatic gradient optimization tool. When dealing with challenging multi-component samples, if a gradient condition doesn't meet the needs of criteria for resolution, then the only option is going to be to try different gradient conditions. Ultimately, this may result in having to re-inject the sample or inject the sample on more than one system with more than one noble phase and stationary phase system. This is obviously inefficient, and what we would hope to achieve is to discover a more sophisticated gradient that may allow us in a single injection to accomplish all of our goals. This is not typically in the past been done by automation, as human intervention is necessary to create the data analysis schedule, and it becomes a challenge to fully automate the process. Rather than engaging in the old way of doing gradient optimization, where a user has to first create an analysis schedule, collect the data, then review the data, and then update all of the methods within the analysis schedule, re-perform the analysis, and then evaluate the data again and continue this cycle until they have reached the optimal conditions. The built-in algorithm within LabSolutions MD performs that data analysis and then improvement of the gradient conditions automatically. By setting the initial conditions and boundary criteria, the software then runs an initial analysis, makes sure that the conditions are satisfied or not, corrects then the analysis, changing it in order to accomplish the desired conditions, and will repeat that cycle until it has reached its goal, without human intervention in between each cycle. And while that may sound sophisticated, the implementation of it is quite simple. Here's a screenshot from LabSolutions MD showing the screen where the user would set the parameters to initiate the gradient optimization process. The flow rate and oven temperature are set, the pattern of the gradient is established, and then, most importantly, the setting of the critical resolution. So in this case, the minimum resolution of 1.5 is desired, and we can choose then the style of gradient curve used for optimization. In this case, it's linear. This will allow, though, still for multiple segments to that linear gradient. Here we see an example of the automatic gradient optimization process, where the top chromatogram corresponds to the initial run of the data. And here we can see that there are two co-eluting species that do not meet the resolution requirement, one earlier in the chromatogram and one later in the chromatogram. As the first iteration of corrective analysis is performed, we are able to get the resolution desired on the first pair, the early eluting pair, but still not quite there on the later eluters. So you can see that the algorithm chooses conditions then for the second corrective analysis, where again we see a change, but not meeting the criteria for both. So the third analysis again modifies those conditions, trying to reach the criteria for both species. And by the fourth analysis, we can see that we're able to get that resolution and separation in a reasonable time frame. So as we wrap up our presentation on LabSolutions MD, I hope you found this information to be useful and you're thinking, how could we incorporate these types of automation tools into our own method development tasks? Because we do design the software around AQbD principles to help you to reduce the amount of time that's spent in method development and to eliminate errors that may occur in the method development process when we try to manipulate all of those manual methods by switching the columns, changing the mobile phases. And when we're evaluating data, the more regular our tools can be and the more they can apply to a large data set, again, we reduce down errors and variability in that analysis process as well. When we combine the technologies of LabSolutions MD with photodiode array data and single quad mass spec data. We have very efficient and simple peak tracking. And using that AI algorithm for optimization of the gradient can allow for very sophisticated gradient conditions to be generated without any user intervention.